हमारा डिसाइड भी नहीं है कि कौन है हमारा
बिंदु मैंने बोला है करके कोई बोल रहा है कोई बोल रहा है नहीं
आना नहीं बोल
क्या फिर बाहर में ना नहीं मैसेज किया नहीं पता नहीं पर आउटर से कुछ बोला किया होता है नोटिफिकेशन किधर no request initiative by did something matter मेरे पापा ने भी तो एवरेज भी कैसे ना निकाला अब यार शॉर्ट स्टेट देर उतना ही तो है क्या क्या हमारे पास के अंदर है जो भी है सॉरी नो प्रॉब्लम
एक और लड़की का बच्चा
how you are doing like this. Now I did it, and I
मंगाया नहीं रिपेयर
क्यों ये टॉपिक वाला वापस बताएगा अभी बोलते ना आगे
कोई आई ना
quality का एग्जाम था quality
शांति आई
किस बारे में एग्जाम फुल ना
क्या देखी लगा लाइफ जब मां को मिला
next page more than that.
मैं साउथ इंडियन में को तभी
अब तो मैं जुड़ गया इसको तो मैंने नहीं आती
जो मैं ले आया
obviously my
एक्मल
तू आज का पार्टी बोले आज का है
मैं तो बस मराठी हूँ
भाई मेरे को बहुत निश्चित है यार निश्चित नहीं ना ये क्या
वो क्या वो ऐसी है कि नहीं बनती है क्या वाटतं ते देता
By the way, for you video you have never is like that my part my series में भी get for so let us series तो मैंने blue से हाँ ये थोड़ा हाँ See, this is gonna be like after after 9 months start we doing amen you देखो बेटा आपकी मस्ती है क्या कैसी मस्ती है प्यार भाई टाइगर में प्यार मुझे समझ गया
आगे तेरा बिल्ली फोटो
ये देख ऑलरेडी प्रिपेयर करके आई आज मैं तो प्रॉमिस कर आज हम फोड़ेंगे
इतना
मार भी मैं तेरे से रामू पर individual bisexual or asexual only sexual the only better sexual ऊपर सिस्टम करना है भाई बस मेरा अच्छी है भाई वो के 3 घंटे में चलेगा अच्छा 30 मिनट के लिए बैठा भाई चार्ली के साथ में हम लोग को मेल पर्सन कैसे नहीं पढ़ा प्रैक्टिकल करके प्रैक्टिकल क्या है अनबन करो
इतना
मुंह मत कराओ श्री राम अबे पेरेंट्स एक्सरसाइज करता
नहीं बट 20% श्योर हो क्या क्या लिखा है ये बात में बता
मेरे को पूछा भाग हां क्या क्या बोला
भाई ट्रॉमेटाइज हो जाएगा वो
क्या बोला
बेचारा पहले पीटीए दिए
एनडी पे भाई लोग का पिक दे वो मैंने नहीं देखा
चार हमने क्या बना उसने सोचा
बेचारा ट्रॉमेटाइज हो
गया पूरा
सॉरी यार हां
तुम लोग की आप सब बनता है मेरे को तो भाई
तुझे कब चिढ़ाया रे तुझे कब बोला तुझे कुछ बोला था मैंने कभी अजीब लग रहा भाई इसका मेरे ये बनाया I like this. I don't know. What is it? I like it. वो मैंने जेनेटिक्स पर मेरे को ये समझ नहीं आया 1 स्क्वायर ओ मोलेकुलर
दिमाग खाती है भाई पे आते आते से टाइम पे करना है
एक तू आया तो कुछ बोलो तू आया तो यार वो मेरे को बोल डाला था पिछला तू तेरे को बोला कुछ नहीं बोल रहा मेरे को बोल के लौड़ी है भाई दिमाग खराब करेगा
क्या वो लाशर पागल बोला रहेगा
ऐसा भाई
अब तू बाहर बैठे हंस रहा था देखो कितना बड़ा बोल रहा है
मेरे को बोल रही है भाई
तुम लोग अरे वो एक्सपीरियंस वाला मेरे को नहीं समझ रहा था लड़की में होने के लिए होना चाहिए लड़के में किसी एक के पास या गवर्नमेंट हो जाएगा ये
नाइस होता है
पर लड़कियों में अगर इनको समझ में आए क्योंकि सेक्स लाइफ किसी तरह भाई के ऊपर तो भाई लड़की को होगा हां लड़की को नहीं होता लड़की का एक्सपीरियंस लड़की का दोनों होना चाहिए तो उससे भी हां उसको बोलो आज क्लास का डायलॉग बना नहीं था मैं क्यों आई हां please don't be like whatever
Why so many absent today? Huh? Test कभी? School में कभी test है? This girl is also absent. What happened to her, Prisha? Prisha नहीं आ रहे? What is psychometric test? In school seminar. Achha. Now, last class what we finished? Genetics. I think last part was there. Haan, only the last topic was there. One thing, just share. You see what are the topics here.
What will we do after this? What will we do after this?
We'll do sex organs and after that we'll study reproductive system. So much to study. Then I'll take some oral. Then I'll give you some questions to solve. So now, hey, no talking. And the last boy can come here. So your head, space to sit. Why do you prefer to sit at the back? So now, we had finished with this part. X-linked inheritance. Let us study this. The other day, we in fact started with this concept. Now, what is X-linked inheritance? We are talking about certain traits which are due to certain alleles present in the X chromosome. We are talking about certain traits. We are talking about certain conditions which happen in human. So, if you remember, the other day I told you that in the male and the female, what is the difference in the 23rd pair? If I talk about the female, in the female, there is a pair of X chromosome. So if I talk about an allele which is responsible for a genetic condition, an allele which we understand that an allele which may be responsible for a genetic condition, but such type of a condition we come across that the males basically suffer from, generally suffer from. It's not that females don't, but very rare. But what is the reason that the females rarely suffer and males most frequently suffer? The answer is that whenever we are talking about the 23rd pair, the X and the Y chromosome, which contains a whole lot of such alleles, and those alleles may be responsible for such condition, genetic condition that we are talking about now. So such allele will always have a Y type known as the dominant type in the other X chromosome. So that's the most common situation that happens in a pair of X chromosome in the female body. So that's the reason this particular allele is not able to suppress, is not able to express. And this particular allele is suppressing this allele, the basic concept of dominant and recessive alleles. But now, if I talk about the X chromosome and the Y, so this is the X chromosome, I understand that this is the Y chromosome. And I said that there is a particular allele which is responsible for any type of a genetic condition. Then what is happening? Is this allele suppressed in the male body? No, in the male body, this allele is not getting suppressed because in the Y chromosome, the dominant counterpart is absent. So in such case, this allele starts getting expressed, even if in a single dose. This allele starts getting expressed, and hence the male now start expressing that type of a condition. This is what we are talking about, a concept of X-linked traits. X-linked traits. Now, these X-linked traits are always found to be, give that attendance sheet. Oh, yeah. Fine. Now, when we are talking about these X-linked traits, again, these X-linked traits constantly keep on passing from one generation to another. And how would they pass on? What would be basically their system of expression, pattern of expression? Their pattern of expression is that now I say that this male is suffering. I consider this as a parent says it. This parent is suffering. Are we going to come across it in the next generation? The daughters, would they suffer? No. With this X chromosome, will the sons have in the next chromosome, in the next generation? So when I understand the F1 or the offsprings here, try to understand, I talk about the offsprings. This X chromosome will always be a part of the daughter's chromosome, daughter's pair of chromosome. So this X chromosome is always inherited. We'll do the process to understand. So this X chromosome will always be a part of the daughter's pair of chromosome, not the son's. So now that means the daughters would basically have a situation something like this, that if they have inherited that, this chromosome they inherit from their mother. So again the daughters are having the trait not able to express because of the dominant trait. So such type of a female is always known as the carrier
Yeah.
Then what about their expression? Their expression is that in the next generation, if I talk about this is the first generation offspring, what about the second generation offspring? This chromosome will again be a part of the male offspring's pair of chromosomes, meaning that again becomes a component of the male offspring in the second generation, not in the first at all. So this will again be a component of the X chromosome, half of the proportions, of course, and then half of them may be having this, and then this allele now again becomes or are present in some male offsprings. Half of the male offspring will have this type of a combination. So again, we come across that if the parents are suffering in the next generation, generally we don't come across offspring suffering because the X chromosome carrying the allele will be always a part of the female offsprings. Then this female offspring will pass it over to their sons again. So that is getting expressed in a crisscross manner, always skipping every one generation. So in one generation, there is a sufferer. In the next generation, there will be no sufferer. In the third generation, again, there will be a sufferer. In the fourth, there will be no. And in the fifth, there will be a sufferer. So this pattern of inheritance, that is what is called crisscross inheritance. Let us see how exactly. With example. Now, in this case, what are we talking about? We try to understand a cross between a normal female. Say suppose I talk about a cross between a normal female. What is the meaning of a normal female? The X chromosomes of the mother or the female does not have the trait at all. I'm not talking about a carrier right now. I'm only talking about a normal female. A normal female. Now this normal female, say suppose marries a colorblind male. Marries a colorblind male. So how do we write down the colorblind allele? Recessive, lowercase letter. What would be the gametes of these type of parents? Female would give rise to only one type. Female would give rise to only one type. So I talk about the gametes. I talk about the gametes. What about the type of gametes the male parent would give rise to? The male parent would now give rise to two types of gametes, which would be this. Half of the chromosome containing this and half of the gametes containing this and half of the gametes containing the one. So I'm talking about what are these? These are the ovum. What are these? These are the sperms, male and female gametes I'm talking about. Let us check the first generation offsprings. We talk about the offsprings now. If this and this fertilizes, what would be the chromosome pair? Capital C, small c. Is this a female offspring? Yes. Is the female offspring suffering? No, the female offspring is not suffering. We already understood that on the other X chromosome there is a dominant allele. So obviously this allele is going to suppress. So what do we write for such type of a female who already has this trait, but we would say that this female is a carrier for the condition that we are talking about, carrier. Then what about this? I talk about this and this coming together, this X carrying over and here. So what would be the pair of chromosomes here? Capital C, and this is the Y chromosome. This is the Y chromosome. What type of offspring I'm talking about? This is a normal female. A normal male, I'm sorry, Y chromosome we are talking about, normal male. So now that we are talking about a normal male, how come normal? Because it's having the dominant illustrative. So we are talking about this is a normal male. This is the normal male. And here we were talking about a carrier female, this was. Let us talk about this gamete now. This and this coming together. What would be the combination? Female again, two X chromosomes. One is this, the other is this one. What about the phenotype? This again becomes a carrier female. This is again a carrier female. What about when we are talking about this and this I took, this and this I took, this and this and this one now. So here I talk about capital C, Y chromosome, normal, male. So this is what should be there in the first generation offsprings. Is there any sufferer? Is there any sufferer in the first generation? No. In the first generation, we will not come across any sufferer, but is there carriers? Yes, the carriers are there. Now, say suppose, if I talk about, if there's a carrier female, this carrier female marries a normal male. Now I talk about, in this generation, F1 offspring, carrier female now enters into a marriage with a normal male. Then we are going to come across what would be the probability. First I want you to, if you have understood, do this in your note. Then we will talk about the second generation. If you have understood, do this in the notebook. If you have a doubt, clarify. First generation parents. After the parents, we are talking about, we come across the parents, their genotypes. Then we talk about the first generation offspring. So fast, fast. Then, क्या हुआ? Urvesh is very hot now. What to say? Urvesh, you have to come and sit in the front or sometime. Right? You may come and sit in the first bench for some time, second bench for some time, cool up and then go back. Continue again with the cross between the carrier female and the normal male. Been late just now. Did you sign here? So if at all you get a 1 mark question, if the parents are normal, if the parents are normal female and colorblind male, do we find any sufferer in the first generation? There will be no sufferer in the first generation. We are going to come across sufferers only in the second generation now. Now continue this once you have completed till here. Now you continue. Let us talk about now the cross, if at all, this way. The carrier female and the normal male. We are not talking about breeding between the siblings or the offsprings, of course, but we are just saying that if at all the carrier female enters into a marriage with a normal male, then what would be the condition? Try now. The second generation. Yes. Okay. And normal male. What should be the type of gametes here? What should be the type of gametes here? This is one type. This is another type. What about the male gametes? This is one type. This is another type. Second generation. The same you can also, I'll give you one example, you will try with the Punnett split. So I'm talking about the gametes, female gametes, male gametes, both are 2 types each. So this gamete, I just number them 1, 2, 3, 4. When I talk about the first female gamete carrying the dominant allele, fertilizing with the male gamete also containing the dominant allele, then what would be the pair of chromosome? The pair of chromosome is going to be something like this. Phenotype would be what? The normal female it is. I talk about cross between 1 and 4. Pair of chromosome would be pair of chromosome would be X and Y, X having the dominant allele. This is again the normal male. 2 and 3. If it is 2 and 3, the pair of chromosome is recessive C, recessive allele, dominant allele, carrier female. This is the carrier female. And now we talk about the last combination, 2 and 4. If it is 2 and 4, then we talk about the recessive allele here and the Y chromosome. What do we talk about this as? This is the sufferer. The sufferer, or we can also write, we are talking about color blindness. So now we have to write colorblind male. So this was the characteristic that we had studied. That if such type of inheritance we study for any type of a genetic trait, what do we come across? We started with one generation. And if in this generation, the trait getting expressed in the male, in the next generation, the trait will be skipped. But the females will carry the allele because the father's X chromosome generally the daughters would get. So yes, that is what is happening in the first generation. But again, if I talk about that, if in the next generation something like this, process of crossing again, something like this happens, then what? Then there is a chance that in the second generation, what should be the percentage of offspring suffering from color blindness now? The second generation? Percentage you have to write. 25%. What will be the percentage of male child suffering from color blindness? 50%. Male child suffering from color blindness, 50%. So these are the typical questions you get in your paper. How I talk about this male on a scale of 4. This male and this male. 100% ho gaya. Half of the males not suffering, half of the males suffering. If the question is, what is the proportion, what is the percentage proportion of offspring suffering? Percentage proportion suffering is 25, because now they are not asking you male or female. If they ask you what is the percentage proportion of male offspring suffering, then you have to take account of only the male, forget about the female. Then out of all the male, 50% has a chance of suffering, another 50% would be normal. What would be the percentage proportion of females that are carriers? Again, 25%. No? Did you understand? So this type. Let us take one more. Carrier, I said percentage proportion of female offspring that are carrier. So I am not talking about this and this anymore now. I'm only talking about the female. So female में से ये तो normal है. Carrier तो खाली 50, half becomes carrier. So 50% I have to write carrier. 50% I said. 25 बोला था मैंने. Okay. So carrier females in the same generation 50. Sufferer males in the same generation 25. Sufferer is 25. Done, no? Take one question, same concept with a Punnett square. Let us do, and I think we are done with this chapter. With a Punnett square, let's do it one. Same question, same concept. Take a question there. What would be the percentage of offspring suffering from hemophilia? Percentage of offspring suffering from hemophilia when percentage of offspring suffering from hemophilia when carrier females marry a normal male. Carrier females marry normal male. Same process में करना है cross. But now after the gametes, you are not to show by this method. You will do the Punnett square. याद है Punnett square? What will you do? How to do the Punnett square? 4 boxes. If you remember, 4 boxes ऐसे बनाओगे. How many gametes are there? 2 gametes per parent. No. So you are writing down one male, one female, ऐसे लिखोगे, and then you will write down both the gametes, write down both the gametes from one parent this side, another parent this side, and do the cross. Genotype, phenotype दोनों ही लिखना है, फटाफट करो. Punnett square. Let me see how many of them remember. हाँ? Female नीचे. अच्छा. You don't remember at all. चलो यहाँ तक करो. After that, I want them to try also. Done. Shina. हाँ? Shina, तुम्हारा knowledge अच्छा है. तुम्हारा presentation बहुत scary.
मैम आंसर बता.
क्या ऐसे you are playing or that cross and zero that way. ऐसे क्या? The examiner will be correcting a paper. In a solid state, you will not be able to see your innocent face. हाँ? So ये presentation देखकर उनको गुस्सा आ जाता है तो you get a loss. जाओ cross फटाफट करो जाओ. फटाफट. ये लिखोगे तो ये कागज है.
कब लिखना?
कितना है बोर्ड में? वो Punnett square बनाना है, तुमने तो बना दिया ना? क्यों?
Punnett square बनाना है.
नेक्स्ट साइड इरेज करो, वहां पे नीटली Punnett square बनाओ, genotype gamete सब लिखना है. This should be a little bit bigger so that you have space to write down the phenotype. Phenotype के नीचे phenotype लिखना है. हाँ, करो. You have a doubt? क्यों? कहाँ पे doubt है बोलो? Write down the gametes. एक parent के gametes लिखो. हाँ, X. Allele क्या है ऊपर? Allele क्या है ऊपर? Allele क्या लिखना है? What are the gametes you will have? Did you get? What are the combinations? Combination क्या था? If it is hemophilic, I gave the same concept I gave. अच्छा, I have done in the form of a chromosome. If you write in the X, same thing. I am writing X, hemophilic, no? So if it is hemophilic, and what I gave you, carrier female. This is a carrier female. This is the normal. What I gave you, normal male, I gave you, no. If it is normal male, then कुछ नहीं आएगा यहाँ पे X, Y, and you may just write it. अब the allele is 0. 0 stands for 0, plus stands for dominant allele. अभी करो. Gametes क्या होगा and how to do it. So accordingly, find out the gametes. What should be the gametes that accordingly you should get here? What would be the gametes? 50% gametes would be this for the female, 50% gametes are this one. For the male, 50% gametes are this and 50% gametes are Y. आया समझ में? What now? Answer लिखो. Percentage for about offspring suffering from hemophilia. So percentage of offspring if asked, 25. Percentage of male offspring if asked, then it becomes 50. और chapter दो में that chapter is right.
ये
हम हमारे में परसों चल गए मतलब हमारे भाई
Percentage means here you have to understand. I'm talking about 4 offsprings. 4 मतलब 100 से 10 में ले लो, जैसे 100. So 4 में से this is 25, this is 25, this is 25, this is 25. So total 100. So how many of them are colorblind? Only this are colorblind, no? 25%. So that's why 25. So if I talk about that, these are also colorblind, then I write 50% are colorblind. That way. Understood? नहीं, क्यों नहीं? कहाँ पे क्या नहीं समझ में आया? क्या नहीं समझ में आया? Now these are the gametes. Okay, I have already written them here. Here. Okay. So what are the gametes now? I'm not going by the crisscross method now. Let us do by the Punnett that he had already finished. So abhi gametes, which are the gametes? 1, 2, 3, 4. No, these are the 2 types of gametes of the female, 2 types of gametes of the male. What did he write here? 2 types of gametes of the female, 2 types of gametes of the male. वही है. Then I talk about अगर मैं 1, 2, 3, 4 इसको करूँ, then this and this is what he is getting. This one. 1 and 3. What about this? 1, 2 and 3. Same तुम crisscross से भी कर सकते हो, but I said do the other method also. So this is done for this gamete done. This gamete left 1 and 4, and 2 and 4 is this one. 2 and 4 normal. 1 and 4 you will get 1 and 4 it is. You are considering 0 as the allele. 0 is considered as the dominant. No, why are you considering 0 as the allele there?
So ma'am, just mark it.
हाँ, तो तुम्हारा allele कहाँ पे लिखा? तुमने allele नहीं लिखा ना? अच्छा, two female gametes. So here it should be the allele. यहाँ पे allele आएगा. So this and this normal. नहीं, carrier कैसे होगा ये? ये carrier नहीं होगा. So this and this would be normal. This would be normal. 1 and then what did you do? This 0 or the last that we take, this is generally the dominant allele that you have to consider. And this you are considering with the 0. 0 or positive. Plus is basically the wild type allele. What is given here in the book? कौन सा पेज बताओ? Wild type 0. Colorblind father. They are considering 0 as a recessive value. What is that? Acha. Okay, I'll do with 0 only. Check. If you have not finished. So here you are okay. If you are considering 0 as the allele, then again I redo the cross again. This is 2 type of gametes of the female, and this is the male. Then this becomes the. But I asked you a cross between normal male, no? Yes. Normal male. So ये हो जाएगा normal male. Gametes X and Y. Punnett square. The female, the male. What are the female gametes? And X. What are the male gametes? So now, those of you have a problem, please check 1 and 3. So this becomes carrier female. I talk about 1 and 4. This becomes hemophilic male. I now talk about 2 and 3, normal female. 2 and 4, 2 and 4, normal male. Now, if I ask you what is the proportion of offspring suffering from hemophilia, your answer is 25%. Okay, now? So this is what you have to talk about: X-linked traits, X-linked inheritance. Now, the last topic, I think, this autosomal inheritance. Autosomal inheritance is also there. Okay, now let us also at the same time talk about the last topic here, that is autosomal inheritance. What is the meaning of autosome? Autosome matlab kya hai? What are autosomes? Autosomes are chromosomes which carry alleles for body characters apart from reproductive characters. Apart from reproductive characters. So now there can be various different such type of autosomal traits. Few examples that are given. Now, if I talk about autosomal traits, can we talk about some of the autosomal traits that all of us we exhibit? What happened? Autosomal traits. Now we talk about autosomal traits. Something like I talk about rolling of the tongue. How many of you can do that? You can do that. You also. Hota hai, wow. Now rolling of the tongue. So when we talk about rolling of the tongue, we talk about the right-handedness and the left-handedness. That's also an autosomal trait. We talk about the hairline, the widow's peak that comes. That is also autosomal trait. Then we talk about the attached lobe or the free lobe like this. A few people will come across that the lobe is not like this. It is almost cut as it is, just straight attached to the face. So the attached lobe, free lobe, joint fingers, 6 fingers. So all these are different types of autosomal traits. Now, just one example that is mentioned in the form of some process, that is, we are talking about rolling of the tongue, autosomal trait that we understand, rolling of the tongue. Now, one such example, autosomal traits. One such example that we are talking about is the tongue roller. Tongue roller. Tongue rolling is because of a dominant or a recessive trait. Dominant. So we may consider that which are some of the genotypes which are responsible for tongue rolling. Which are some of the genotypes? If the chromosomes have allele known as capital R, that is designated as capital R, yes, the person may show tongue rolling. The person may show tongue rolling. But is it necessary the person has to be only homozygous for this allele? No, the person can be heterozygous. Then also the person would be tongue roller. When would the person be non-tongue rolling? The person would be a non-tongue roller only when the person has a recessive genotype. So these are sometimes you are asked also what are some of the genotypes for a person who is a tongue roller. A person who is a tongue roller can either have a genotype something like this or have a genotype something like this. Can we understand that generation after generation whether the trait tongue roller will be expressed? In what proportion will it be expressed? Yes, if we are talking about a cross between such type of parents, we talk about a cross between such type of parents, then we can easily understand what would be the proportion of inheritance of such type of a trait. And if we talk about inheritance in a number of generations, 2, 3, 4, 5 generations, this is what we are talking about as pedigree analysis. This is what is called pedigree analysis, meaning inheritance of a particular trait we are trying to study in few generations all throughout. So the same trait we try to see how it is expressed and in what generation it is not expressed. So that's a small topic that was left here. Look into some questions, just few questions. You must have already done this in the school. Look into some questions that I want you to try. I'll give you some questions here in the book. You don't have too many questions also. Number C, sub number 4. देखो, number C, sub number 4. See that? Number C, sub number 4. You try that first. Try that first. Number C, I said sub number 4, page number फटाफट करो, number 35, page 35. Explain why generally only the male child suffers from color blindness and not the female. Just 2 lines answer to write. What will you write there? बोलो बोलो हाँ तो बोलो ना. You are raising your.
A male individual has only one X chromosome, and if this X chromosome bears the recessive affected allele.
कहाँ से तुम पढ़ कहाँ से रहे हो?
The symptoms of the disease are exhibited. There is no corresponding allele on the Y chromosome, whereas in a female there are 2 chromosomes. There are 2 X chromosomes. A female is affected.
रीडिंग भी वो गलत कर रहा है. कुछ भी नहीं समझ में आया. Look at that question. Number 4. Explain why generally only the male child suffers from color blindness. What is the answer? You should write in 2 lines. Just now we had done those process. Why should only the male child suffer from color blindness? Because the male or the X chromosome, when in the X chromosome of the male, the recessive allele, when in the X chromosome of the male, the recessive allele for color blindness, when in the X chromosome of the male, the recessive allele for color blindness, is present, the trait or the allele is expressed. The allele is expressed if in the X chromosome of the male, the recessive allele for color blindness is present, comma, the allele gets expressed. The allele gets expressed. Full stop. This is because the Y chromosome do not have. This is because the Y chromosome do not have. The Y chromosome do not have any contrasting dominant allele. Y chromosome does not have any contrasting dominant allele. Look into the volume. In the volume, there are some extra questions. Let us just take 5 minutes to solve some questions, and then we move on to the next chapter. Take some 5 minutes to solve some questions. Look into your volume, all of you. What happened now? Chalo, fast. Look into your volume. Do you have your volume with you? No, you cannot bring. Now, from this chapter, let us try some questions. Look into page 42. 42. Fill in the blanks, karo, fatafat. 42, fill in the blanks. And MCQ. 42, fill in the blanks, MCQ. Complete this. There's definitions and all are simple, not to worry. Fill in the blanks and MCQs karo. Species. No, that part is not there. Chromosome number you are not asked. Chromosome. 38. Only human you know, and at the most you are asked about plants like onion, 2 pairs, only that much. 2. 8 pairs. 8 pairs. 8 pairs, huh? Lion, this is just 19 pairs, 38, that's it. Only this 3, 4. This hardly comes. This doesn't come. Why it's not happening now? Done. Allosomes are sex chromosomes, 1 pair. MCQs. I just asked you to do this so that you get a fair idea. Otherwise, the process are not able to practice at home. Karo, 5 minutes mein karo, and then we go to the next chapter, sense organs. Then break. Break I'll give you later. We'll have to finish up some portion, otherwise there's no time. You're starting. Sense organs. We'll complete with sense organs. Let's see. And then still after that, what we have to run. Just 5 minutes to take.
I mean, it's like
Johan. In some books, you will get written John. That's not correct. Johan is Johann.
Maybe there is a question in which year did Lieberman discover certain phenomena that cover transmission of hereditary characters in the garden?
1904. 1904.
1906.
Okay.
18
अरे एक्सिट का
दिया है ना
हम क्या
Options है homework and classwork. Few questions you do so that you get to know. Don't take too long, huh?
An exception of Mendel's law and humans.
What is it?
An exception of Mendel's law.
Exception of Mendel's law is what is this given codominance? Link.
Dominant purity of gametes lie and
लिंकेज ऑप्शन है कि कोडोमिनेंस कोडोमिनेंस अगर है तो वो भी करेक्ट होगा लिंकेज है तो वो भी करेक्ट होगा डोमिनेंस इज देयर प्योरिटी ऑफ gametes is there segregation is there independent assortment is there if you get an option linkage that is not there in his concepts It's 1865.
1865 है
1865. That is power day. It's okay. That is not a very important question. That is not a very important question. Also, only to find on that. Chalo, finish card. Done. Solve some questions there. Now, we are already completed with the concept. Done, no? All of you could solve some questions. The rest, there's crosses which are there from the volume. Some crosses are there. Whatever questions you would basically expect from your object is obviously many. And secondly, some crosses you may expect. Punnett space you may expect. Crosses where you will straightaway be asked about explanation of the laws, dominance and segregation and independent assortment. For independent assortment, you are generally not allowed to or generally not asked to do the cross on the paper because it's quite lengthy. But the cross will be there on the paper and questions on it. So you should have a fair idea about the cross, also the ratios on it. Now, the basic concept of nervous system. To again talk about the basic concept of nervous system, already you know what are neurons, what are the different types of neurons, sensory, motor, mixed, all these basic concepts of neurons you already know. Now we talk about function of neurons. What did we understand? Carry messages to the CNS. CNS contains what? What are the two parts of the CNS? Brain, spinal cord. So today we're going to talk about that part. The brain and spinal cord. We are talking about the brain and the spinal cord. So under sense organs, what are we studying? Under sense organs, we will be majorly talking about the structure of the human eye and the ear. So let us talk about the human eye first. What are the parts of the human eye? So we talk about the parts of the human eye. to start with this sense organ, what are some of the functions? When we talk about what are the functions of the human eye, the functions of the human eye to understand helps in the perception of light, helps in vision. So first of all, when we look into that, what would be some of the characteristics of the human eye? Before we look into the, can you see the other side? Or it's appearing blurred for you that side? You can't see. But this suffers from spondylitis a lot. That's the problem. So now that we are talking about the structure of the human eye, what are we supposed to first study? Accessory structures before we talk about the main parts. Accessory protective structures. What are some of the accessory protective structures? Remember about the human eye, I'm first of all talking about some accessory structures. What are some of the accessory structures? These are majorly helping in protection, not truly vision. What are some of them? Let us go one by one. We talk about the eyelids. We talk about the eyelashes, the eyebrows. Now, if I talk about the eyelids and the eyelashes and the eyebrows, more or less the same function that we understand. What are they majorly helping the eye in? How is it protecting the eye? Not allowing unwanted substances to fall on the eyeball, the outer lining of the eye. So that is one function. Not allowing unwanted chemical substances, water droplets, unnecessary to fall into the eye. So these are actually functions. We don't understand much. So ye ho gaya, some of the major characteristics. Apart from this, what else we get to see associated with the structure of the eye? Apart from this, we also come across the lacrimal glands. Now that is important here. So if I talk about the tear glands, lacrimal glands, so one that is very important to understand here would be the lacrimal glands, and this lacrimal glands release what? It releases tears, and one component of the tear is an enzyme known as lysozyme. Take a note of this. We talk about the lysozyme. What is lysozyme? It is a digestive enzyme, hydrolytic enzyme, we can understand, which majorly helps in the microbes, if at all, on the surface of the eye. No, if at all, there are some germs on the surface of the eye. So that is the job. Apart from that, the tears, what would it generally help in? Clearing the eye. So that much only you know. What else? The eye that we understand is actually lodged in a cavity. It is lodged in a bony space inside. No, so that this entire bony socket, that is what is known as the orbit. So the bone that we talk about is actually the extension of the skull. So it is actually the extension of the skull that we are talking about, and hollow. And this eye is attached to that part with the help of some muscles. Some 6 pairs of muscles are there. Each pair of muscle has got one or the other job to do. So this 6 pairs of muscles, underline karo, 6 pairs of muscles. How many pairs of muscles? 6 pairs of muscles are there. And these 6 pairs of muscles actually help us in the movement of the eye in different direction. Front, or we talk about the upward movement, the downward movement, and the oblique. Okay, the oblique, 2 different types of oblique movement in an angle that we talk about, the oblique movements. So these type of movements have been brought about by the eye muscles. So that is about the accessory structure to understand. Now we look into the layers of the eyeball. Now that's a very important part of the structure to understand. Layers. Now, what are the layers of the eyeball? To talk about now the most important, there is a constant noise in the class. तुमको मैंने इतना अच्छी तरह से समझाया था, फिर भी तुमको नहीं समझा। मैंने एक शब्द नहीं कहा। Then who is talking? Shri Ram is talking. Tanmay, Soham, you are talking. No. Then the walls are talking. Layers of the eyeball. What are the different layers we are going to study? We are going to talk about, first of all, the sclera or the sclerotic. Second layer is choroid. And the third layer would be the retina. Let us learn. So first of all, we are talking about the layers of the eyeball. Kitne layers hai? Again. Kitne layers hai? 3 layers. Primary layers of the eyeball. And these are the 3 layers which are helping in vision. So now let us talk about the first layer that very clearly in the figure you get to see. The sclera or the choroid. What are some of the properties you should remember? It's made up of fibrous tissues, white fibrous tissues, no blood vessels, no pigments at all. It does not, or the overall layer is opaque. The overall layer is thick, dense, opaque actually. But this layer, if you see here, it takes a bulge in the front of the eye. The white portion of the eye that we get to see is this particular layer that we're talking about, made up of white fibrous tissues and no blood vessels. No pigments, again to remember. So this is the sclera that I'm talking about. This is the sclera that I'm talking about. But in the front of the eye, the sclera takes a bulge. In the front of the eye, the sclera takes a bulge, and all throughout it is dense and opaque, but here in the front it becomes transparent. So these are the features. So what is this bulge known as? What is this convex structure known as? The sclera is extending in the front of the eye to form what is known as cornea. The sclera is extending in the front of the eye, becoming convex, becoming curved, becoming transparent to form what is known as cornea. So now that I talk about that, what are some of the properties of the cornea? It is transparent, it is convex. What are some of the functions? Significance kya hai? Significance yahi hai that since it is transparent, it is allowing the light to pass through. And since it is convex, it is also allowing the light to get converged in. So I said that ye jo convexity hai is actually allowing the light to enter and then get converged to fall at a particular point on the eye. We'll learn what is that. So now that I talk about the light falling on our eye, every time now that the light is falling on our eye, is it entering in one particular direction? In various directions. No, I look at the book, the light is hitting the book and then the reflected light is falling here. And from any part, the moment I'm looking into any particular object, every time the light from different directions are entering into our eye. The reflected light rays from different objects, from different directions are entering into our eye. But हमारे eye में अगर कोई specific structure ऐसा नहीं है जो light को एक particular direction में अंदर ले जाए, then the vision would not have been possible. So this is the first structure that we are talking about. The convexity of the cornea is allowing light यहाँ से आएगा and in every particular direction, but this light should be converged to or made in a conical arrangement. The light rays should move so that the light falls on a particular region in the inner layers. So that is about the cornea to understand. Does the cornea have any pigment? Does it have blood vessels? Nothing. We talk about the second layer, choroid. What is the second layer? Listen carefully. After that, I'll give you some questions to answer. Choroid, what I said, that's the second layer. Dekho. This is the second layer. Can you see the light pink region there? In your book also, mark the choroid. The light pink region that you get to see. Very close to the choroid, very close to the sclera. So now this sclera is there. Now next to the sclera, we come across a light pinkish shade. The light pinkish layer that is the choroid. What are some of the major properties that you should remember? Again, number one, I'm saying the choroid is pinkish because it has got rich supply of blood vessels. The choroid has rich supply of blood vessels. Unlike the sclera, the choroid has a large number of or large deposits of pigment granules. Which pigments? Melanin. Which pigments? Melanin pigment is scattered but. So now I said two major properties that you have to remember. What are the two properties? Choroid has rich supply of blood vessels. Choroid has pigment granules. Which type of pigment? Protein. They are all protein in nature. Which pigment? But melanin pigment. So these are the two major properties to understand. But now I again look into the extensions of the choroid. Look into the extensions of the choroid. As the choroid moves forward in the front of the eye, the choroid in both the regions, at the posterior and the anterior. At the upper region, at the lower region, the choroid is taking a bulge to form something known as the ciliary body. So what can I say? That this choroid actually takes a bulge to form another structure. What is this other structure known as? This other structure is known as the ciliary body. Ciliary body is an extension of the choroid. Can you see the ciliary body in your diagram also? The ciliary body is there. So what is ciliary body? It is just a mass of group of muscles. It is just a small swollen structure made up of muscles. What type of muscles? Circular muscles. What type of muscles? Remember these names. Circular muscles. What type of muscles? Circular muscles would be there. Where are circular muscles present? Circular muscles are present in the ciliary body. So I said this choroid extends in the front of the eye to form the ciliary body. Bolo. AC. It will be so suffocating without the AC. You are feeling cold. You. Put off the AC. Okay, wait, I'll call him. Wait. So now that we talk about the choroid, what to understand, what to remember, the choroid extends in the front of the eye, first to form the ciliary body. Extensions of the ciliary body on both sides, the upper and the lower, the anterior and the posterior both. But this ciliary body, what did I tell you? What type of muscles does it have? Circular muscles. You will be asked in the paper. What is the function of the circular muscles? These circular muscles can contract and relax. And this, can you see in the figure also, these circular muscles are actually attached to this part. The circular muscles, these circular muscles are actually attached to the lens. Check in your figure, check the diagram that you have also in your book. Can you see the circular muscles are attached to the ciliary body? Let me just show you another diagram. See here. The ciliary muscles are. Here it is there, but these fibers, of course, you can't see that clearly. Can you see these fibers very clearly here in this figure? Can you see these fibers very clearly? It is something like, when I talk about the lens, this is the lens there. And if I talk about the ciliary body, first of all, the extension will be something like this, and then the ciliary body would be here. And this ciliary body will be connected to some fine thread-like structures. It will be connected by some fine thread-like structures. So if I talk about, say, suppose the lens here. Now this is the lens, and I'm saying that this is the ciliary body going there, and then there will be the pupil running after that. And these are some fine thread-like structures, and this is the same with the other side also. Some fine thread-like structures, some fine fibrous tissues are there. So this is the lens I'm talking about. What is this part? I said this is the ciliary body, and this is an extension of choroid. And we are going to learn later that this is again an extension of the choroid known as the iris. So first of all, if I talk about the second layer of the eyeball, the second layer of the eyeball extends in the front something like this. And this is the ciliary body. This ciliary body remains connected with this fine thread-like structure, which are known as the suspensory ligaments. So first of all, these suspensory ligaments would constantly be pulled and relaxed. These suspensory ligaments will be pulled at times and they will be relaxed at times. So contraction and relaxation constantly happens, and this is what is changing the shape of the lens. This is what is changing the shape of the lens. So this lens, which if it is normally like this, biconvex, sometimes the lens can become bulged like this. And sometimes the lens can even become comparatively lengthened and long like this. So the lens is at times becoming lengthened like this, or sometimes becoming extensively bulged and shorter also, based on how we keep on focusing. So we are focusing on near objects, we try to see something nearer, the lens is trying to become shorter. We try to see something far away, the lens is becoming long and extensive and long. So what is happening? The convexity of the lens is progressively changing as we change our focus. So which part of the eye is responsible for this? Ciliary body containing the circular muscles which regulate the movement of the suspensory ligaments.
Suspensory ligament is just holding the lens
In position, suspensory ligament holds the lens in position. That is a function that you are asked also. But whenever you are asked, name the part of the eye which is responsible for changing the shape of the lens, then though we understand that the suspensory ligaments playing a role, but it is majorly the ciliary muscles playing a role, and then the suspensory ligaments. Am I clear here? Am I clear? So what did we understand? First of all, that one part, one extension of the choroid. What is one extension of the choroid? Ek to hai ciliary body. What is the other extension of the choroid? The other extension of the choroid is the iris. The other extension of the choroid is iris. So what are some of the properties now to understand? I said just now that the choroid extends in the front of the eye to form the ciliary body. The ciliary body has got the circular muscles, and further this choroid extends on either side of the lens, anterior and posterior both. Can you see the small finger-like projections, almost little bit overlapping on the lens, little bit covering the lens. So this small finger-like projection that we talk about, this is what is known as the iris. Now this iris has got two types of muscles. This iris has got two types of muscles. One is known as the radial and the other is known as the circular. Two types of muscles the iris has. One type of muscle known as the radial, the other is known as circular. So if I now ask you, write one difference between the iris and the ciliary body or ciliary body, what is the main difference that you are writing? The ciliary body is swollen, iris is basically tubular finger-like projection, and the muscular differences are one of the prominent difference that you should write. What I told you just now, iris has radial and circular both. And what type of muscles are the ciliary body has? Only circular muscles. Okay. Now here, what are some of the differences just now we had learned between the choroid and the sclera? Sclera is white fibrous, no pigment, no blood vessels. And choroid, just now what did I tell you? Choroid has large number of blood vessels, pigment granules. What about the iris? Iris also has got melanin granules. And the presence of the melanin granules in different proportion is what is responsible for the color of the eyeball. So we talk about the color of the eyeball. What are the prominent colors with respect to genetics? Also we had learned prominent colors would be the brown and the black and the blue. So the brown and the black and the blue that we are talking about, the prominent colors of the eyeball, this is perfectly because of the pigment melanin present in the iris. So name the part which is responsible for the color of the eyeball. When you are asked, you are supposed to write iris. And iris is an extension of what? Choroid. Yes or no? So we are talking about, we had already talked about two parts. Which are the two parts we talked about? The sclera, the first layer. Sclera extends to form cornea. Sclera extends to form cornea. The sclera extends to form the cornea. Remember. And now I'm talking about the choroid. Choroid first of all extends to form ciliary body. The choroid first of all extends to form the ciliary body, and then this iris. Choroid extends to form the ciliary body and then the iris on either side of the eye. Now, since we are talking about the iris, something more that we try understanding is, say, suppose I'm talking about that this is the iris on the other side, and this is the iris on the other side. The ciliary body goes like these other layers. So now I'm understanding that the ciliary body is there on both the sides, and this is the iris. Is it not that the iris is actually covering the lens partially? The iris is partially covering the lens, but still there is a space left for the entry of the light rays. So I'm saying that this iris is partially covering the lens, but this space which is left between the two iris will be termed as the pupil. So what is pupil? The gap between the iris in front of the eye. The gap between the iris in front of the eye is known as the pupil, which is responsible for the transfer of the light rays, which is responsible for the passing of the light rays, so that the light rays can enter into the inner portions of the eye. So this is about the pupil. Can we see the pupil in our eye? When you look into yourself in the mirror, when you look into yourself, your eye in the mirror, then you can see a small circular structure. Exactly. You can see a small circular structure and muscles radiating out. So that is how the pupil looks. Exactly. When we are talking about the structure, the pupil would generally look like this. The pupil would generally appear like this. What is happening with this girl? Why don't you sit this side also? This side would be better also, no? Band karna ki, can we reduce, increase the temperature a little bit? Increase the temperature a little bit. The room will be too hot. It's already 26. 25 it will be cooler. ठीक है, थोड़ा देर बंद कर दो. After that, we'll like to put it. What happened? What is it? क्या हुआ? क्या हुआ? क्या प्रॉब्लम है? प्रॉब्लम क्या है? What happened? Sit there for some time, not a problem. Sit there. Now what? What is this? Huh? We'll complete with this, then I'll give you a break, not to worry. Okay, now next is the third layer. So about the sclera, about the choroid. When I ask you questions, about the sclera, about the choroid. Take one question now. You're going to write. Take one question. Write two differences. Notebook, all of you. Put the head of the chapter, sense organs, the first question. Write two differences. In the paper, you will be asked conditions. Now, I'm not asking you condition. You have to write differences based on their composition, position. Write two differences between sclera and choroid. Two differences between sclera and choroid. So I said already the major two differences you have to write on is their composition and their pigments. So this is the major point that you will be asked also. Composition pe likhoge, so what are you supposed to write? Sclera made up of white fibrous tissue, does not contain blood vessels and pigments. Choroid consists of blood vessels, rich supply of blood vessels and melanin pigments. Second point, you will write about the extensions also now. Since melanin pigment I already told you as the first point only. Extensions likhna hai. Extensions, what will you write? Sclera extends in the front to form cornea and iris. Dono likhna hai. Extends to form iris and ciliary body. Likho fast. Done. Third layer that we are to study would be the retina. I'll complete with this and then I'll give you a break. Don't panic. Now the third layer that we talk about would be retina. Retina is the innermost layer of the eyeball and the most important layer of the eyeball. Now look into the retina there. I talk about retina as the innermost layer of the eyeball and the most important layer of the eyeball. So what are some of the properties of the retina that you are supposed to remember? Retina has got photoreceptor cells. The retina has got large number of photoreceptors. Photoreceptors. Which are the photoreceptors? We talk about the rods and the cones. So we are talking about two types of photoreceptors, the rods and the cones. Let us try to understand some major differences between the rods and the cones. Now, when we look into a detailed structure of the retina, just to show you the detailed structure of the retina majorly appears like this. It is a complete nervous layer made up of bipolar neurons. So when I talk about the retina, the entire layer would have only arrangement of neurons all throughout. So now this photoreceptor cells that we are talking about, what are some of the differences that you are supposed to understand? I said, if I talk about the rods, the rods are scattered all throughout the retina. The rods are scattered all throughout the retina. Meaning, if I talk about the rods and the entire retina, the rods are almost scattered all throughout the retina, and in no region of the retina they are concentrated at one particular region. But that is not the concept for cones. When I talk about the cones are concentrated at one particular region, and this particular region is known as the yellow spot, also known as the fovea. Yellow spot, also known as the fovea centralis. Let us see, where is the fovea centralis? So I talk about the first difference that I'm talking about is that if you look into this particular small depression on the retina, can you see in the diagram in your book also? There's a small depression on the retina, almost on the horizontal axis of the eyeball. There's a small depression on the retina. What is this depression on the retina known as? Yellow spot, also known as fovea centralis. Remember the terms: fovea, macular lutea. That's also the other term. So macular lutea hai, ya to fovea centralis hai. So one is the same thing. So now that we are talking about what is fovea centralis, macular lutea, we are talking about a region on the retina where the cones are concentrated more. What about the rods? They are more or less scattered. But at the same time, you have to also remember that if I talk about retina as a layer, now say suppose I'm talking about that this is the choroid, and I talk about that this is the retina. If I understand that this is the retina layer, always try understanding that if I understand the retina more or less at the periphery, the retina is more or less arranged at the periphery. And where would the cones be present? The rods, if they are more or less at the periphery, and if I understand what about the cones would be majorly concentrated at the fovea. Again, I said, if I talk about the entire fovea, there will be a small depression. There will be a small depression, and this particular depression is what we are talking about as the yellow spot. I talk about the entire region. This is known as the yellow spot. So if I'm saying that this is the yellow spot, a particular region on the retina, then I should write that the rods are more or less on the periphery and the cones would be maximally arranged near the yellow spot like this. The cones would be maximally arranged this way. So this is one concept to remember. I said the first point of difference is whether they are scattered, where they are present. Next, what type of pigments are they producing? The rods are producing a visual purple pigment known as the rhodopsin, and it exists only in one form. Only in one form it exists. But if I have to talk about the cones parallelly, the cones are releasing another type of a pigment known as iodopsin, which comes in or which is present in three forms. Three forms. Cones are also present in three types. Rods are present in only one type, and the rods are producing only one type of pigment, rhodopsin. Cones are present. How many types of cones? Three types of cones, and hence three types of iodopsin also. And why three types of iodopsin we are talking about? Because of the three primary types of color vision. What are the three primary colors we are talking about? Three primary colors are what? Blue. So now these three primary colors that we are talking about can be perceived by the three different types of cones, three different types of iodopsin. And how do we see the other colors? Because most of these colors are combinations of different colors or different shades. So when we are talking about rhodopsin and iodopsin, you are asked in the paper how many types of iodopsin? Three types and one type. Next is, let us talk about their sensitivity. When we are talking about their sensitivity, these rods, they can produce rhodopsin in a very dim light. What do you understand by the sensitivity here? I'm saying that whenever we are talking about sensitivity, we should understand sensitive to dim light. What is the meaning of dim light here? Very low intensities of light. Very low intensities of light. Something like, if I now talk about bright light here, perhaps will not be able to see anything sharp. वो sharp focus हमें नहीं मिलेगा. Can I put on the fan?
Yes, yes.
This fan? No?
Yes.
It's very hot to me.
Yes.
So now, when we are talking about sensitive to dim light, so when I say sensitive to dim light, what do you understand? Rhodopsin will be formed only when light intensity is low, and for black and white vision, what is the meaning of black and white vision? To see any object at the same distance, sharp focus, same size, correct orientation, without any color. That is what is called black and white vision. So black and white vision के लिए do we need a very bright light? No, bright light होगा तो we will not be able to see. So dim light होगा then the focus is clear. The process of image formation is efficient. What about the other side? But if I talk about iodopsin, here I should write they are very much sensitive to bright light. Sensitive to bright light, meaning whenever I talk about formation of the efficiency of iodopsin and perceiving color, can we perceive colors in a low intensity of light? No, it's difficult. We cannot. Even if our own room, our own house, which we every day are acquainted with, even then, say suppose you are in your room and there's a power cut. After a few minutes, you are at least able to see the objects, the chair and the table and the bed, but are you able to see the color perfectly of the curtain and the cushion cover and the bedspread? No, you are not. Why like that? Because intensity of light, when it is low, it will not give us clear vision about the color. What else about the rods and cones you are supposed to understand? Rods help us in black and white image, black and white perception of image, and cones help us in color vision. Cones help us in color vision. So now when we are talking about the degeneration and the regeneration concept, what is the meaning of degeneration? I talk about. What are we to understand by the concept of degeneration and regeneration of these pigments? Let us talk about degeneration. मतलब degeneration का simple meaning क्या है? Degeneration का मतलब है breakdown हो जाना, bleach हो जाना, मतलब inactive हो जाना. It's a protein. And regeneration का मतलब फिर से बनना, again getting formed. So if I have to talk about rhodopsin, rhodopsin will be formed, or if I start with degeneration, rhodopsin will be broken down if the light is dazzling. Rhodopsin will be broken down, the pigment will be broken down, inactive हो जाएगा if the light is a dazzling light. So do you have experience when the light is too bright, you can't see anything very sharp? No, because rhodopsin is becoming non-functional at that time. But now the intensity of light is adequate, and इससे कम भी होगा अगर light है also we are able to see. Why? Because regeneration of rhodopsin. Rhodopsin can be formed at that intensities. What about the other side? For the cones, it will be just the opposite. For the cones, when the intensity of light is low, then will the iodopsin be formed? No, they are inactive. At a higher illumination only, the cones will be releasing iodopsin adequately. So what are the 5 prominent differences we had been talking about just now? What are the 5 main differences I told you? First of all, if I talk about the rods and the cones, number 1, what are the rods meant for? That's their function. You have to remember always. You will be getting questions. Rods and cones bracket mein function based on the pigment, based on their location, based on their sensitivity, etc., etc. So all these points you should remember. So what I said again, rods majorly meant for black and white vision. For cones, color vision. What about the location? I said in the retina all over, at the periphery or scattered all over the retina. And what about the cones? Fovea, mostly concentrated near the fovea. And what about its sensitivity to light? If it is low intensity dim light, which type of pigments are active? Rhodopsin. Iodopsin is it active? Iodopsin is not active. At a higher intensity of light, iodopsin is active, but rhodopsin is bleached, degenerated. And what type of, how many type of cells? rods only one type, cones 3 types. So these are some of the major differences between the rods and the cones that you talk about. After, when we talk about the specific function of the retina, if you're asked in the paper, write the specific function. What is the most important function of the retina? What are you supposed to write? Retina is that particular layer of the eyeball which helps in image formation. We'll talk about the process of image formation later. After the break, we'll again continue with the structure of the eye. We are yet to talk about the chambers of the eyeball, the lens, and certain more basic structure. Take a break. You. 15 minutes break. Question break is complete. Question break is complete.
छोटी बात साढ़े 7:29 को भी नहीं छोड़े अबे पर टाइम नहीं
Yeah.